Okay, so good afternoon, everyone. Hello, welcome to this session about nanomaterials and nanotechnology. It's really a great pleasure to have you all here. I think we have a very interesting session with kind of a broad subjects. I mean, we're talking about a lot of different things. A few general notes. This session is going to be recorded to the request of the organizers. If any one of the speakers does not want the session that we record his talk, so just please send me in the chat and I will stop the recording. I apologize in advance that I won't be able to introduce all the speakers in the really appropriate form because of due to time. So that will be kind of short. If you can turn your cameras on, it's always nice to have more people that you can see while we talk, so that can make this more interesting. If you have any questions, feel free to send them in the chat or ask at the end of each talk. So without further ado, we'll start. We're going to start with a keynote speaker, Professor IVS-IPSTA, and then we'll have a couple of talks, and then another keynote speaker. At the end, Professor Origami, and then we're going to have a roundtable discussion. I'm going to send the question that we're going to raise before that discussion. I already sent it once. So let's first have Professor IVS-IPSTA from the Weizmann Institute. I will not be able to tell you about all his scientific honors because they span over 5 decades, depending how you look at it, maybe even 6. So, but I will tell you that he's a huge promoter of science and collaboration through prizes that he organizes and international organizations. So I think everybody knows him around here. And we're all thankful to him for all his contributions. So, Reshef,
Hagai, toda rabba. Thank you very much, Hagai. And I'm sharing with you my screen. What do I see? Oh no, sorry. What happened? Do you see? Okay. So. What's going on? I know. Okay, so let me go straight ahead to the topic. I will talk to you about inorganic nanotubes and fullerene-like nanoparticles, a topic that follows that I'm following or started 30 years ago. In fact, I'm the oldest person, probably the oldest person working with 2D materials on Earth. At least for 37 years, I'm working day and night on only on 2D materials, and this is my focus of interest. And I will start from 2 general slides or historical slides. So I want to show you one of the earliest slides on works on 2D materials. This is the work of Linus Pauling from almost 100 years ago. This is the first work on molybdenum disulfide structure. And you can note from this slide that he was also the first one to realize that there are layered structure, 2D materials, what we call today 2D materials, or layered structures materials. And that he was the first person to cleave these materials, which leads us immediately to the opportunity to get them layer by layer as we know today. I won't go into details of this work, wonderful work. As you know, Linus Pauling was a very smart scientist, and he concluded a lot of things from this work. But I want to show you the next slide, and I'll go rapidly on that. This is another work of Linus Pauling, which is a benchmark in a sense for nanotubes, because this is the first work describing nanotubes. In our modern language, nanotubes are based on layered or 2D materials. And here he looked at the chrysotile and kaolinite. These are 2D materials, which you can see they are made from infinite layers with silica in the bottom and alumina at the top, connected via vicinal oxygen atoms. And this is an infinite layer. And what he showed in this first paragraph, what he said was that using X-ray, you could determine that these materials will form nanotubes. He was visionary because at that time, one could not even see nanotubes. There was no electron microscope which could see nanotubes of this size. And indeed, it took another 20 years to see that. And but the other statement that he did was a mistake. He said that molybdenum disulfide, which is stated here, won't be able to make nanotubes because it's a non-asymmetric. He distinguished between asymmetric compound with small base at the bottom and large base in the top, to mica, for example, which is symmetric, has the same base at the top and the bottom. And he said that since molybdenum disulfide belongs to this section, they will not form nanotubes. And he was, of course, wrong, but for a different reason. But he established the first mechanism for formation of nanotubes using asymmetry of layered compounds. And the second mechanism, which is known today more or less as Croton-Ijima-Tene mechanism, was the fact that these materials suffer from edge effects. So I won't go to the details, but as you know, C60 was discovered in 1984 or 85. And the reason that it forms was due to the dangling bones in the graphite or in the graphene sheet, which are very reactive, and they tend to form either full C60 or nanotubes. And so using this analogy to molybdenum disulfide, I claimed that, and I proved that layered compounds like molybdenum disulfide and tungsten disulfide, which are the archetypes of layered compounds, inorganic layered compound, will also form nanotubes and fullerene-like structures because they suffer from the edge effect similar to graphite. And with that notion in mind, I went to the lab to prepare these nanostructures. And here you see the first generic nanostructure there, and nanoballs or spherical fullerene-like nanoparticles. Unlike C60, they are not made of one layer, single layer, they are made of many layers, 15, 10 layers, and they are large, relatively large, 20, 30, 40 nanometers, and containing millions of atoms, but nevertheless, they are all closed cage structures. And this is important for many of their properties. And at the beginning, we couldn't make them we could see them in very, very small amounts. And nowadays, of course, we can synthesize them in pure phases, and we can distinguish in their physical properties from the bulk materials in any physical measurement that you do. But we had first to learn how to synthesize them, and that took us quite a long time to understand. So these are the nanotubes, the typical tungsten disulfide nanotube taken with the best microscope available 25 years ago in Oxford. You see the multi-layers, and you see that they are well arranged. And if you go 10 years ago, 15 years ago, there were already the CS corrected microscope, and you could see the atoms. The atoms are clearly there. Sorry. You see the tungsten atoms and the sulfur atoms, and you see how perfect they are arranged. And you can see also, you get the impression of how much nanotubes, pure phases of nanotubes we can make. And this is quite an accomplishment. And I will go directly to the recent work of Professor Alazak from HIT. And she was able to synthesize, after many years, also molybdenum disulfide nanotubes. So first, we synthesized tungsten disulfide nanotubes in the year 2000. We were able to form them from tungsten oxide and synthesize them in a way that they first grow nanowhiskers, then they sulfidize on the surface, and slowly the sulfide replaces the oxide core, and we get a nanotube. She did it also for molybdenum disulfide. This is a remarkable work, but here, because molybdenum oxide is not stable as a nanowhisker, she had to go to different temperatures. She did it at different temperatures and different flow regimes. So this work was actually the first time that macroscopic amount of molybdenum disulfide were obtained in a process that can be scalable. And now I'll go immediately to the mechanical properties of nanotubes. And why are they so remarkable? Because they are, if you look on the unusual map of materials, they are either strong or they can be elongated. They cannot have the both properties, namely high strength and high fracture toughness. And with the nanotubes, you can have both of them at the same time, and you can see that from the work of Yifat Kaplan-Ashiri, how the nanotubes are strong and flexible, and they don't fail. And this is important for many, many applications, mostly for polymer nanocomposites, which are now investigated in great detail and are on the way for industrial application, major industrial applications. But I won't go into that in detail because I don't have the time. I want to show you just one application as an example before I go to the rest of the talk. And this is the plates that were prepared for protection for safety jackets. So here is a plate made from epoxy fabric, aramid fabric with phenolic resin. And this is without any nanotubes. And this is with 1% nanotubes. And this laboratory experiment in Serbia, in Belgrade, they could show that the bullet, when they shoot the bullet penetrates the blank or the neat plate without the nanotubes, and it does not go through the plate, even though there is only 1% nanotube there, and you can verify it here, how the bullet starts to penetrate, and eventually it is stopped here. And then amazingly, the mechanical properties of the nanotubes and their large surface area and their dispersibility in the polymer allows them to stop very efficiently the bullets. So let me go now directly into the optical properties of the nanotube. This is our recent work. This is a recent work with Iwasa's group in Japan, where they showed that these nanotubes exhibit bulk photovoltaic effect. What is bulk photovoltaic effect? In the usual photovoltaic effect, we know that the effect is produced by the junction between a metal and a semiconductor, or semiconductor and P semiconductor. Here, in the case of a nanotube between two electrodes, the electrical current, the photocurrent, is in the middle. It's not close to any junction, and it is a quantum mechanical phenomena which emerges from the toroidal motion of the excitons or the electrons through the nanotube, which produce a kind of an electromagnetic field which push the nanotubes to the electrons to the left and the holes to the right, and you produce a photocurrent. And this is quite a remarkable work that showed that one can get a very high, relatively high bulk photovoltaic effect by this in nanotubes, not in plates, not in single layers of tungsten disulfide. Let me go to another work. This is a work that was initiated by Dr. Lena Yadgarov. She is now an assistant professor in Ariel University.
5 minutes,
I beg pardon?
5 minutes.
5 minutes, okay, so I would skip it. Then I'll skip it. Oh, it's really fast. Then I'll skip it. So I'll go directly into the recent work of misfit compound, nanotubes for misfit compound. Misfit compounds are compounds which are in many ways similar to chrysotile, namely they are asymmetric. They are made of intercalation of lead sulfide, for example, in between two layers of tantalum disulfide, and you get a completely rock salt lead sulfide in between two layers of tantalum disulfide, and these are completely these are intercalated layers. They are asymmetric, and consequently we assumed that they will form easily nanotubes, and they did. We realized it. The process that we use several processes, but the one that we favored was VLS, and we hit the material with catalyzed, and eventually we collect the nanotubes at the cold end of the furnace. And here are pictures of such nanotubes. You see clearly the structure of the rock salt. This is the nanotubes. By the way, you see the nanotube. This is the rock salt, the lanthanum sulfide in this case, and the tantalum disulfide, very well arranged nanotubes, and we could make them from many, many misfit compounds. And this is a cross section of such compound, and we can bisect them with a FIB, and you see perfectly the lanthanum atom and the sulfur atom, or in this case it's a samarium atom, also the tantalum and the sulfur atoms. So actually, there is a recent work that has been published this year with my student Sridhara. He gave a poster, presented a poster on that. We could make them asymmetric, namely the lanthanum is bound to sulfur only, and the tantalum is bound to selenium. By making the right proportion between selenium and lanthanum, we can make them very asymmetric with a huge dipole moment. And if we went from 0.2 selenium to 0.8 selenium, we could see that they modify their structure completely. Instead of having lanthanum sulfide, tantalum, samarium sulfide, tantalum diselenide, they went to samarium sulfide and 2 layers of tantalum disulfide. And this is clearly seen here. And this is even further clear in this bisection or this lamella, which shows you the beautiful structure of this tantalum disulfide, lanthanum sulfide, and so repeatedly. And we actually now investigate their properties. We are slowly gathering more information on their unique properties. Among other things, they are superconductors, and they have other interesting properties. And with that, I would like to conclude basically by saying that we showed in our work that many layered compounds, 2D materials, are actually, if you choose the right chemistry, and usually it's a high temperature chemistry, you can make from them nanotubes, and you can make if you can make them smartly, you can get interesting properties which are different from the bulk properties and different applications afterwards. And that's actually what we have shown. We still are confined to tungsten disulfide in the case of application, but nevertheless, the number of application expands from quite substantially in recent years. And with that, I would conclude my talk and will be happy to answer questions.
Thank you very much, Reshef. Excellent talk, very interesting as always. Questions, please. Anyone? Okay, I have a question, Reshef. So you showed a lot of progress in different aspects. Going forward, if you have to choose one thing that that is like the top priority to improve, what would you go for? Or what are you working on now?
Synthesize more nanotubes from different layered compounds, understand the structure, and elucidate their properties. This is for me the first priority. And the second priority is applications. And by the way, it is done through many collaborations, which, because I didn't have time to mark them, I just put them in as the publications at the bottom. We have many applications. A new paper that is being submitted now is on a new neuromorphic device that can function. I cannot talk a lot about that, but can function as a memory device, basically.
Okay, thank you very much. Very interesting. Any more questions? Okay, so before we move to our next speaker, I would like to share with you, I just sent in the chat, the question that we're going to talk about in the roundtable discussion at the end. There are going to be panelists that will address these questions first, but then everyone is encouraged to participate as well. Okay, so we're right on time for our next speaker, Professor Gil Markovich from Tel Aviv University. Gil? Yeah, so
Hi.
Gil, hi. So Gil did his postdoc with Jim Heath in the previous millennia. And since 1998 has been at Tel Aviv University doing a lot of interesting things in nanomaterials and nanotechnology. And he's going to talk about now symmetry breaking in the formation of chiral nanostructures. Gil, bevakasha.
Do you see my presentation?
It's kind of stuck. So we see.
Yeah, it's stuck with me too. Let me try again. Oh my God, it's stuck. I will try to start again the PowerPoint. So I will just start speaking, and while it's coming up, we work in the recent years a lot on nanoscale chirality, different aspects, and let me see. And today I will tell you about one of these systems that we study. And now I'll try again. Great. You see it?
Yes, looks good.
Okay. So, so I guess I don't need to introduce chirality very much, right? It's just the absence of mirror symmetry, or the fact that an object that is cannot be superimposed on its mirror image. It can be molecules, it can be nanoscale object, or even a macroscopic object like this sea snails. We are interested in nanoscale objects because they are, if they are smaller or of the order of the wavelength of light, so they can do all kinds of chiroptical effects, that is, affect the polarization of light passing through them. So in this introduction, I just refer to Louis Pasteur's work, right? Because he crystallized salts of tartaric acid, which fortunately crystallized as two separate enantiomers, and he could observe that the crystals themselves, the macroscopic crystals of the two enantiomers, had asymmetric facets, and so he could distinguish between the two types of left-handed and right-handed crystals and dissolve them back and observe that each of them rotates the polarization of light different directions. And we were interested a few years ago in doing similar stuff, but we were interested in doing similar stuff, but with inorganic materials where the building blocks themselves are achiral. For example, like quartz, right? You have SiO2 and the building blocks are achiral, but they can crystallize and form chiral crystals. So they can be either right-handed or left-handed. All these chiral crystals have some helical motif in one of the directions of the crystal. And so the crystallization process breaks the symmetry and can make them either right-handed or left-handed. And of course, in nature, they should appear at equal proportions, the two-handedness. Why is it interesting? So, you know, you can have different considerations. One of them is the whole pharmaceutical industry and biomolecules, which are, of course, it's important to get them enantiomerically pure. Part of the interest in symmetry breaking in the formation of chiral materials comes from trying to understand origin of life, and particularly the riddle of homochirality, how homochirality in amino acids and nucleic acid and sugars and so on came to be. And even in just the problem of crystal nucleation and growth, looking at chiral crystals and looking at the moment of, let's say, decision of when they decide to become right-handed or left-handed would tell us probably a lot on how crystal nucleation occurs, which is, in spite of being studied for so long, still not well understood. So in this particular project, the target is trying to control how much, let's say, if we can cause all the crystals to become left-handed or right-handed, and how can we probe the crystals and learn about the proportion of left-handed versus right-handed crystals, nanocrystals that we have. So the system that we have been studying in the last years and turned out to be very interesting is one representative out of a whole group of lanthanide salts called also rhabdophanes, So we're working particularly with nanocrystals of terbium phosphate hydrate. We dope them with a little bit of europium, which can perfectly replace terbium because they have very similar chemistry. They crystallize in the same space group as quartz. And the idea was, let's try to use some chiral molecules in the presence in the synthesis of the nanocrystals to try to affect the handedness of the formed crystals. So we needed to choose some molecules that we know that should interact strongly with the components of the crystal. So we chose tartaric acid, the same molecule that Pasteur used, because it has these carboxylate groups that like to complex strongly with these trivalent lanthanide ions. So that was the system, and it makes very easily, you get needle-shaped nanocrystals from this material in synthesis in water. So the idea here is that you excite the terbium in the UV and it transfers energy to the europium ions. And the europium ions are very good reporters of chirality. So they should have some preferential circular polarization emitted in at least in part of the emission lines. It has several emission lines. So by filtering out the difference in left and right-handed circular polarization emitted from the europium, we can get an idea about the ratio between left-handed and right-handed nanocrystals that we form, because it should be proportional to that. And there are very many advantages of using lanthanide-based chemistry for this kind of experiments. We don't have time to go into it. So the synthesis, as I said, very simple. You just take the components, the phosphate and the lanthanide ions. We do it at acidic pH of 2. And the synthesis, we started initially to work in boiling water, 100 degrees C, and I will also show you results for lower temperatures. And indeed, it worked. So the tartaric acid molecules seem to guide at least the ratio between left-handed and right-handed nanocrystals to be unequal. Depending if you take D or L tartaric acid, you can get opposite circularly polarized emission spectra. This is the intensity difference between the two circular polarizations, what you see here. And if you take a racemic tartaric acid, that is equal proportions of the tartaric acid and enantiomers themselves, you get this yellow line, which is 0. So initially, everything seemed to be working fine, and it was very easy to detect this circular polarization because of this efficient europium reporter of the chirality. Now, of course, the first thing we went to check is tartaric acid unique in that sense? So it turns out that it is quite unique. If you take glutamic acid, which is very similar to tartaric, it's chiral and it has 2 carboxylates too. So you get much smaller effect in terms of optical activity, which indicates that the difference in the enantiomers of the crystals is also much smaller. And another molecule that is very similar, also chiral to carboxylates, called camphoric acid, 0 effect, nothing. So clearly the whole issue is about the interaction of the chiral molecules with the building blocks of the nanocrystals. So that was one part of the study, the initial part of the study. Then the even more interesting part was the following graph. So here on the horizontal axis, you have the tartaric acid enantiomeric axis. 0 means that you have a racemate. As you saw before, it gives you 0 optical activity. G-lam is this ratio. Here you see it below. It's the ratio between this difference in the intensity divided by total intensity, which is basically a magnitude of the indicator of the magnitude of optical activity. So you can see that as we go towards pure enantiomeric tartaric acid, of course, you get optical activity, which means that you break the symmetry, but it saturates at relatively low value. So already when you have like 40% enantiomeric excess of tartaric acid, which is about 70 to 30% right to left or the opposite. So you get saturation of the optical activity. So to us,
5 minutes?
No problem. So to us, this was an indication that maybe in this case, we get this saturation indicates that we get 100% enantiomeric selection of the nanocrystals. And as I will show you in a moment, we were able to prove it using a different technique. Just to say something more about the different molecules. So for example, when you take the glutamic acid, which has this very weak effect on the handedness directing of the nanocrystals, you see that when you change H here is this, again, the enantiomeric excess of the tartaric acid, you get this sublinear increase to a very small value compared to the saturating behavior in tartaric acid. Later on, we also studied other molecules even more similar to tartaric acid. For example, malic acid is really similar to tartaric acid, except for one OH group missing. In malic acid, actually, you can get also complete enantiomeric selection when you have pure L or D malic acid, but it grows much more slowly as a function of the enantiomeric excess of the molecules. And actually, if you take lactic acid, which is half of tartaric acid, just one chiral center with only one carboxylate and one hydroxyl, you get basically almost very negligible enantiomeric selection. So it means what we learned from this and other molecules that we tried is that actually you probably need three functional groups that will bind to the building blocks or to the surface of the evolving crystals in order to be efficiently guiding the handedness of the nanocrystals. So just to finish, we have very shortly to prove that we had 100% enantiomeric selection in these samples using the tartaric acid. We collaborated with the group of Ori Cheshnovsky, who does all kinds of fancy microspectroscopy experiments. In this case, so we did CPL, circularly polarized luminescence experiments on single nanocrystals using basically a fluorescence microscope, a home-built fluorescence microscope that was equipped with circular polarization filtration. And here you can see, for example, the difference in emission spectra of a single nanocrystal of the two circular polarizations. So you can see that at particular lines, you can see very big differences between the two. So those can be used to determine if a single particle is either right-handed or left-handed. We actually use the machine learning algorithm to overcome signal-to-noise ratio issues. So this was even more efficient. So we could define or determine the handedness of single particles with the confidence level of over 99%. And indeed proved that the nanocrystals in those samples were enantiomerically pure, except for one single particle in one of the syntheses that we actually discovered that it's the opposite enantiomer. But let's say on the 99% level, they were all the same nanocrystals. Then we could actually take TEM grids with nanocrystals on them and then determine each nanocrystal if it's right-handed or left-handed, and actually repeat the Pasteur experiment on the nanoscale using electron and optical microscopy combined. So just for the last thing, when you go down in the temperature of the synthesis, 50 degrees and 40 degrees, so you see that these curves, this saturation curve becomes sharper and sharper until, let's say, even at 40 degrees, it's even very pronounced, that even without an enantiomeric preference of the tartaric acid or in the total absence of tartaric acid, you get breaking of symmetry. So this is like spontaneous symmetry breaking, or it's actually almost spontaneous symmetry breaking. So very tiny impurities of one enantiomer will cause the whole system to break the symmetry. So this is something we are now trying to understand. So just to summarize, we believe that this is a very interesting model system to study and understand this kind of symmetry breaking effects that occur also in chiral molecules sometimes, and even maybe help us to understand other symmetry breaking problems, like in the one related to the origin of life. And also in general, we believe that this kind of studies will help us to study and understand better nucleation and growth of nanocrystals in general. So thank you very much for your attention.
Thank you very much, Gil. Toda. Questions, please.
Gil, can I ask you a question?
Sure.
Is it possible to use Raman, polarized Raman or polarized IR to define the chirality of your crystals?
Yes, this is a very good question. We're actually collaborating. There is a technique called Raman optical activity, which is actually it's very tough instrument to build. So there's only one company that makes them, or people build them themselves. And indeed, we started observing, actually not in this system, although maybe it's possible also in this system, but in chiral mercury sulfide, that you have basically chiral phonons, and you can, so you can observe the chiral phonons, and this can also be used to determine the handedness of the nanocrystal. That's an excellent question, actually. Thank you.
Any more questions? If not, we'll thank Gil again. Thank you very much, Gil.
Thanks.
And we'll go to our next invited speaker, Tamar Segel-Perez from the Technion. Tamar is a very special speaker today because she is this year's, she just won the prize of the IVS Early Career Award. So let's all congratulate Tamar. She will be also formally acknowledged during the last session of today. At the end, please all stay and congratulate her again. So Tamar did her PhD with the GTFI, the Technion, then moved on to do her postdoc with Paul Neely at the University of Chicago and Oregon National Lab. Since 2016, she's at the Technion. Tamar, bevakasha.
Thank you, Hagai. Can you hear me? Okay, so thank you, Hagai, for the kind introduction and for inviting me, and I'm very happy to share with you some of my group's research. We mainly focus on combining polymers with atomic layer deposition technology for functional materials, and today I'm going to show some of our work regarding using that for constructing membranes and improving membranes for water filtration. So as you probably know, water stress, water scarcity, providing clean and abundant water for the growing population of the world is one of the major challenges for humanity in the 21st century. And membranes, especially polymer membranes, play a central role in a solution, or many solutions for this challenge. So why do we need ALD and all that? So before I explain why, let's just briefly introduce ALD if you're not familiar with this technique. So atomic layer deposition is a part of the chemical vapor deposition technology. It enables very precise thin films deposition using sequential exposure to vapor precursors. So you can see in this classic example of ALD growth of alumina, the precision and the sub-nanometer control over thickness comes from the fact that the reaction is divided into two half reactions or two half cycles. Each of them is self-limiting. Okay, so an organometallic precursor is absorbed on the surface, and later we introduce a co-reactant. In this case, it's 2-methyl aluminum and water, creating a monolayer of alumina, and then this is repeated to achieve the desired thickness. And this is a mature technology used quite extensively in the semiconductor industry. It has diverse inorganic chemistry, it's solvent-free. And for membranes, it's important because it's uniform and conformal deposition on high aspect ratio and tortuous structures. And although ALD is mature, mainly it's been used for inorganic materials growth on inorganic substrates. And only in the last decade or so, people started to ask what happens when you do ALD on polymers. So today I'm going to show you two types of ALD on polymers and specific on polymer membranes. One is classic ALD, but on polymers. And the other one is ALD within polymers. So what is called sequential infiltration synthesis. So we drive the precursor into the polymer and grow the inorganic material. So let's start with ALD on polymers. You see here an example of an ultrafiltration membrane. So ultrafiltration is used to filter bacteria, particles, in the high end also viruses. This is prepared by phase inversion. And if we apply ALD on this pristine polymer membranes, you can see from the high resolution cross-section TM that we can form a very thin inorganic metal oxide layer on the membrane. In this case, it's 2.7 nanometer. And it's very uniform and conformal, creating now a new interface between the polymer and the water environment. So this enables us to do two main things. First, this is very precise. So we can turn that to precise control over the pore size. You can see here two examples, polyacrylonitrile membrane and polyetheramide membrane. Both of them are around 20 nanometer average size in their pristine membrane. And then with additional ALD cycles, as you can see here, we can reduce the pore size even to sub 10 nanometer region. With that, we have also a price to pay. Porosity is lower, so there's a trade-off, and the decision is based on the exact application of them. The second thing, of course, is we change the interface. This is we put a metal oxide, such as alumina, we enhance the membrane hydrophilicity. And with this enhanced hydrophilicity, there's a reduction in fouling, for example, in oil fouling. So you see here confocal microscopy of all water emulsion. The oil droplets are in red. And from this confocal microscopy, we can deduce what is the surface coverage with time and with flow. So polymer membranes is almost completely covered with oil fouling, while the hybrid membrane, where the interface is not metal oxide, we see a reduction in surface coverage, and everything is washed away quite easily when we apply just pure water in the cell, the flow cell. Okay, so far I've discussed ALD on regular commercially available membranes such as phase inverted membrane. You can also find quite easily in the industry track etched membrane. As you can see, both of them are not ideal. Phase inverted membranes have large pore dispersity, while track etched membranes have low pore density. We would like to combine the advantages of both of them. And this can be done using self-assembly of polymers. So you see here a block copolymer structure. And due to the fact that block copolymers are made out of two or more distinct blocks, and their dissimilarity drives their assembly into highly ordered structure. In membranes, we typically target the cylindrical phase. We can create a high pore density and uniform pore size structures. And this can even be further improved if we now combine in them inorganic materials. And that we do with this process called sequential infiltration synthesis, an ALD derivative that enables the inorganic materials growth to occur within the polymer. Basically, we use high partial pressure and long exposure time of the precursors to drive them into the polymer. And we judiciously choose a polymer that will react with the precursors, creating hybrid inorganic materials with nanoscale precision. As you can see from this example of a high resolution scanning DM image of zinc oxide clusters and particles in PMMA film. And this, when combined with block copolymers, give us a lot of possibilities because we can choose the block copolymer chemistry and the precursor chemistry to create selective growth in only one block, creating unique hybrid materials. And if we want, we can later remove the polymer and template inorganic nanostructures from our block copolymer structure. So for example, we can take a block copolymer that assembles into cylindrical film. This is non-porous yet. We grow selectively alumina in one of the domains, the blue domain in this case, it's polymethyl methacrylate, remove the polymer, and then obtain highly porous inorganic membrane. You see how uniform the pore size and the pore density. And this is now templated by our initial block copolymer structure. So we've shown that you can use that to separate protein, but the truth is it's not very scalable when you do it in thin films. A better method to use block copolymers is to use what's called asymmetric block copolymer membranes. So the entire structure here is made out of block copolymers. Our collaborator in Germany developed and manufactured this kind of membrane. So it's one material, but it's fabricated in a two-step process. First, you cast the membrane, let it evaporate a bit. So this drives the self-assembly and the ordered structure on the top. And then immerse it in non-solvent, and this drives porosity and the creation of the supporting substructure, which is spongy. And these are very efficient ultrafiltration membrane with pore size targeting ultrafiltration. We asked if we can use our methods to drive it to the very challenging nanofiltration regime, where you can actually filter out pharmaceutical compounds in water or toxic small nanoparticles. And the idea here is to take the porous Glucopolymer structure and add inorganic material using SIS and ALD, controlling the pore size, but also creating a surface that is susceptible for small organic functionalization, creating tailored nanofiltration.
5 minutes, Tamar.
Great. So as I said, this is in collaboration with Professor Volker Abetz from Helmholtz Zentrum in Heeren, Germany. So we start from a Glucopolymer system. This is polystyrene, poly-4-vinylpyridine. And polystyrene is quite inert to the SIS process, while P4VP is very reactive with the organometallic precursor. And we see here two examples of two different Glucopolymer systems with different pore size. And with just 3 cycles of SIS, we grow so much alumina in the P4VP, so it swells, and we get a significant reduction in pore size. Then if we want to further reduce the pore size, we do that with additional ALD cycles. And eventually we can start with an easy membrane fabricated from large Glucopolymer molecules and drive it down to small pore size. And as I showed you earlier, SIS and ALD are very conformal processes. So the first few microns of the membrane is functionalized by the ALD and SIS process. Okay, and then we have now a new surface, not just with the interaction of the water, between the membrane and water, but also for additional functionalization. So with a luminous surface, we can do a quite easy wet process of silanization and add small molecules on the surface, creating specific functionalization such as fluorination, cationic functionalization, or anionic functionalization. With this functionalization, the membrane structure is still preserved. The water permeance is reduced as soon as we reduce the pore size and add functionalization, but we gain the nanofiltration aspect. Okay, so for example, if we take 2 molecules that are very similar in size, but slightly rose-bengal is more hydrophilic, than ruthenium chloride complex. If we just use the Glucopolymer with alumina, we don't see a big difference, or it's not enough to really retain one and separate and let go of the other. But with the fluorination functionalization, we can retain almost to 100% rose-bengal, while the other one is passed through the membrane. And we can do that with other functionalization. Here we have 4 dyes. 3 of them are negatively charged, 1 is neutral, and there are size between 1 to 2 nanometer. And we can separate them both on using charge selectivity and size selectivity, containing or retaining one of them while passing away the other. So this is really an array of abilities where we can tune both pore size and functionality to create the desired filtration. And with this, I'll summarize. I've shown you that the ALD is a very powerful technique for membrane surface engineering. SIS can be used to construct membranes and to change the surface, creating hybrid organic, inorganic membranes with specific functionalities. I'd like to thank first my students and the people who really did the work, especially Asaf Simon, Tamar Itzhak, and Maya Barzilai, who've seen some of their work here today, funding, and you for listening.
Thank you very much, Tamar. So now we all know why your prize is well deserved. Thank you.
Thank you.
Questions, please.
I have a question or suggestion. I don't know. maybe it's not so relevant because in the last part of your talk, you had pores, but I guess they had a very small thickness, right? Below them, they had larger volume. But if you had like narrower, longer pores, so your narrowing of the pores technique with the ALD basically could also help you to do some sort of size focusing of the pores, because I would imagine that if you work under special conditions in the ALD, maybe not reaching real equilibrium, but being flow controlled, so you could maybe reach more uniform sizes while narrowing, because the small ones will not be so much coated like the larger one, right? Is it something?
Yes, and we've been investigating that. So 2 comments on your comment and suggestion. First, in membranes, we do want to preserve this kind of thin selective layer, because then the diffusion through the large pores is not limiting. What is limiting the selectivity is just the thin layer. In terms of the physics of growth, you're absolutely correct. And especially in polymers where the diffusion coefficients are quite low, we utilize that in order to create gradients. And some of our work, we already investigated that, and we're now in process of doing that in fibers and films. And you can actually grow specific components in different locations, create three-dimensional structure in pores and in other architectures. And it's a very, very interesting technique.
Okay, actually, I have a few questions, but due to the time, I will ask you offline afterwards, if that's okay. So
yeah,
thanks again, Tamar. Thank you. And now we're going to move to 2 contributed talks. The first one by Sujit Kumar from Time Group, associated with the Weizmann Institute and Bar-Ilan University. Sujit.
Thank you, Professor Spiceman. I will share my screen. Can you hear me?
Yes, we hear you loud and clear.
Okay, good. Good afternoon, everyone. I'm Sujit Kumar, currently working as a postdoc in the Weizmann Institute of Science and with David Kahan. Today, for this episode of the IVS conference, I would like to present our results on the atomic scale insights to the 2D on 3D halide perovskite interfaces. This work has been done in collaboration with Lothar Hauben and Katja Reckhoff from the electron microscopy here in Weizmann, and of course with David Kahan. So what are halide perovskites? For optoelectronic applications, the class of the perovskite that are of particular interest and have shown some amazing optoelectronic properties are the lead-based halide perovskites. The crystal structures of these material consist of a framework of a corner-sharing lead halide octahedrons, and the void in between them generally is occupied by a small monovalent cations like methylammonium or formaminidium or cesium. Similarly, there is a subclass of these perovskite which are called layered halide perovskites, or the 2-dimensional halide perovskites, which were once highly explored in 1990s for their quantum confinement effect. These layered perovskites can be formed by literally slicing this 3D structure along their inorganic planes, and this is typically done by using a bulkier or a larger A size cation, and this can be typically of phenylethylammonium or butylammonium, or many such examples are there. So recently, scientists have been exploring the opportunity to use these layered perovskites on top of this 3D structure in order to have, or in order to achieve better device efficiencies and stabilities. And the results have been quite promising indeed. If one makes a device with a 2D, a few nanometer of 2D on top of a 3D perovskite structure, one generally consistently reports an improvement in the power conversion efficiency due to the improvement in the open circuit voltage. And not only that, the major improvement is seen when the devices are more stable even under the ambient conditions. So there exists a clear interest in exploring this 2D on 3D halide perovskite architectures. However, till now, very little knowledge have been gained regarding their interfaces. And most of the results till now are based on the photoluminescence or XRD measurements, which are a bulk or macroscopic measurements. And the results have shown that these interfaces found between the 2D and the 3D perovskite are quite dynamic. That means they undergo some structural rearrangements by movement of certain species. And as a result, this pure 2D and 3D interface is converted into a mixed 2D and 3D perovskite interface. So it would be nice if we could have a nanoscale picture of what is happening at the interface. For such a requirement, generally it is preferred to have a transmission electron microscopy studies. But for perovskite, which is highly beam sensitive, this is a big limitation. And as you can see, these material have a very low beam damage threshold. And within electron dose as low as 100 electron per angstrom square, the material starts to degrade and form some non-stoichiometric perovskite phases. And basically, the material what we wanted to study is not the same as once we shine the electron beams on top of it. To overcome these beam sensitivity issues of the perovskite, Lothar, our collaborator, and we came up with an idea that we will use this 4-dimensional scanning transmission electron microscope coupled with this nano beam electron diffraction measurements. The results of these measurements I'll be showing you subsequently in my next slides. But before that, for preparing the samples of this 2D, 3D perovskite, we wanted basically a phase pure 2D on top of a 3D perovskite. We used a standard recipe first for making NaPbI3, which is our 3D perovskite structures, looks like this, using the precursors material that we have. And then we used a vapor phase surface cation exchange by using these 2D precursors in order to have a conversion of the top few nanometers of the 3D into a 2D structures. Our XRD does show that we have a different crystallographic phase at characterized by a lower diffraction peak at a lower diffraction angle, and which is related to the higher D-spacing of the new 2-dimensional perovskite that we have made. Similarly, the PL measurements have shown that in addition to the 760 nanometer peak, which corresponds to the 3D emission, we have a new emission at around 520 nanometer, which comes from the 2-dimensional perovskite that we made by our process. And basically, there is no any other emission peaks, which shows that we have a pure phase of 2D and a pure phase of a 3D material. So now coming to this 4D STEM experiment that I briefly talked about previously, here we show a typical 4D STEM experiment that was measured on our 2D, 3D perovskite samples that were carefully prepared by Katya under FIB microscope. And in this technique, what happens is you have a few nanometer convergent electron beam that raster scans the sample across, and for each sample position or the probe position on the sample, we can record this diffraction pattern, which stores all the crystallographic information from the sample for that particular location. In one particular scan, we typically acquire thousands of them, and this is done within few seconds, and with one diffraction pattern which is captured in just 1 millisecond. So
3 minutes? 3 minutes?
Okay. So all being said, how does this 2D and 3D perovskite looks at a nanoscale? So what you see here is, and the bottom panel, what you see here is a virtual bright field image that we have constructed by mapping the non-deflected beam intensity as a function of this probe position. As you can see from the contrast differences, we can easily distinguish the silicon, the perovskite, and the carbon core. However, that the 2D that we have made on top of the 3D perovskite is not quite easily distinguishable. But since we have all the crystallographic information from each point of the sample as the electron beam scans across the sample, we can actually map for the occurrence of the 2D lattice spacing, that on which locations the 2D lattice spacing occurs. Here in this virtual map, what we see is the bright domains are reflecting the 2D phase that grows on top of the 3D perovskite. And this can be also confirmed by seeing the diffraction patterns that are locally averaged from a region in 3D perovskite and from the surface region, which supposedly should have 2D perovskite on top of it. Then we also wanted to study how these material behave under or evolve under the continuous electron beam irradiation. For that, we actually choose a sample and then continuously illuminated the sample region and continuously acquired the electron diffraction pattern, which is taken in each millisecond time frame. And as you can see, the diffraction patterns, the higher order diffraction pattern rapidly vanish. That means the 2D films are not very stable under the electron beam irradiation, and the higher order reflections are vanishing. That means we are losing the long range ordering. And not only that, when we repeatedly scan the samples under the E-beam, we could see these white domains which reflected the presence of 2D phase is actually disappearing. And not only that, we also see some extra two-dimensional extra diffraction spots appearing, which reflects to the formation of quasi 2D phase that was till now inferred from the photoluminescence and XRD measurements. With this, I would like to summarize my presentation. What we could achieve is that we could show that the suitability of a 4D STEM experiment and coupled with a nanobeam electron diffraction experiment can tell us, can help us to study this beam sensitive material in their state of interest, and that is without any degradation. And under continuous electron beam exposure and under repetitive E-beam scans, we could show how, or we could establish the time scales in which the 2D phases lose their long range ordering, and also we could show evidence for the formation of quasi 2D phases. With that, I will thank my collaborators, Lothar, Katya, and David Kahin, and Professor Hongxing Chen from Zhejiang University for contributing to this work. Thank you.
Thank you, Sujit. Thank you very much. Interesting. Questions, please? I have a question. You said the 2D structure under E-beam loses its integrity. Well, I mean, the 3D structure, is it stable under E-beam?
The 3D structure, surprisingly, we found that since we have actually used a 4D STEM technique, which normally people don't use, because this 4D STEM technique should give us more ability to study this beam sensitive material. However, this 2D material has not been yet studied, and we could establish the time scales in which the 2D materials are losing this integrity, but we didn't find any such phenomena occurring for the 3D perovskite with our measurement conditions.
Okay, thank you very much. So let's thank Sujit again. Thank you.
Thank you.
And I would like to ask our last very special guest for today is Professor Oleg Gan from Columbia University. Boker Tov, Oleg.
Boker Tov, Hagai.
Yeah, so Oleg is now in the West Coast. The time there is now what? 5 in the morning?
4:45, right?
4:45, yeah, just about before 5. Did you at least have coffee?
No, it's okay. I hope the talk will gonna wake me up. So
okay. So Oleg completed his PhD with Moshe Deutsch, and then moved on to do a postdoc at Harvard University. After that, he was a faculty at Brookhaven National Labs for like 14 years, and then from 2016 he's at Columbia University. Oleg, bevakasha.
Toda rabah, Hagai. Thank you for inviting me, and I will be happy to share our research. So we'll discuss programmable assembly of nanoscale architectures. You can see my screen well, right?
Yes.
Okay. So let me just basically to tell you why we're doing what we're doing. So we all know how materials and variety of structures built on variety of scales, which is shown here on the left side, from very large one down to nanoscales, using more or less top-down approaches. So conceptually, if you have a blueprint, there are different ways to deal with the blueprint and convert it into reality. The problem, however, as soon as we go into the very small scale, it becomes intrinsically very, very difficult to create three-dimensional structures. And so the most advanced approaches in lithography are typically well suited for two-dimensional or layered structures, but not for three-dimensional. The difficulty becomes even greater if we consider how we can utilize a variety of nanoscale blocks which available now and have been developed in the last couple of decades in form of nanoparticles of inorganic materials, which comes in different materials, different shapes, different ligand functionalization, and so on. It's not limited only to nanoparticles. We would like somehow to use also either natural or de novo proteins and build materials from them. So from this point of view, the challenge is that if we establish a platform where we not only can study why self-assembly happens in a particular structure, but deliberately drive system into the targeted structures according to our design. And that's essentially the problem we're trying to address. So the way how we address this problem is asking this question that we can rely on some tuning of conventional interactions for example, of electrostatics or maybe van der Waals and so on, but typically it doesn't give you enough freedom really to create structure completely by design. And the approach we're using is utilize the power of DNA in its ability to prescribe interactions in a highly programmable fashion, where the interactions provided DNA strands which can be encoded to find each other. So I wanted to stress that unlike conventional interactions, let's say physical interactions, where typically kind of binary, you have negative, positive charge, or you have or you don't have interactions like for example, van der Waals or gravitational. DNA also gives us the ability to differentiate different interactions through the encoding. For example, two particles, as shown here, will recognize each other if they have complementary DNA, but they will not recognize each other if they don't have complementary DNA attached to them. And this can be happening in a pool of other millions of other particles which might have a different type of DNA. So this kind of ability to introduce what we call colored interactions, it's quite unique, and I will show that this potentially opens possibility really to encode complex organizational structures. However, if we're going to go and even discuss this question, how we build structures from those nanoparticles using DNA, of course, we want to first understand even the simplest case scenario, and the simplest where we have, for example, many DNAs attached to the particle surface and they're forming shell. I want to stress this is already complex enough, unlike this type of scenario where you have here, where you have only one type of particle, one DNA connecting two particles. When you have multiples, there are a variety of effects associated with polymer physics will emerge in this type of scenario. And going from this kind of hybridization of two DNA to asking how I can prescribe the formation of, let's say, three-dimensional superlattice become challenging. So some time ago, quite a long time ago, we showed that there are different ways to tune interparticle interactions when particle surfaces are coated with DNA. And so one of the ways which is actually successful in our ability to regulate those interactions and form well-defined superlattices is shown here. So we have a DNAs attached to the particle, even though it's shown only one strand. In reality, we have, for example, for 10 nanometer gold particles, it will be around 50 DNAs attached. And the idea is here that we can use interparticle, we can change interparticle potential by changing this kind of attraction part, which is through the recognition, which is shown kind of as a colored, versus spacer. And spacer essentially does provide us ability to tune entropic repulsion between the particles. And so by playing with those two parameters, we can essentially be in a very different regimes in terms of interparticle interactions. And it turns out that we need to be in this kind of intermediate regime. This is kinetically not favorable to obtain superlattice. This is simply the system is not too soft to get to the crystallization regime. We can form well-defined structures. I wanted to stress, and this is kind of from equilibrium point of view, in addition, it's very important to have crystallization pathway, and crystallization pathway essentially means that we can tune temperature and achieve a proper wafer system to transfer from disorganized but assembled into the crystalline. And so in order to do this, we can in situ monitor the formation of the system using small angle scattering, which is coming through the sample, produce pattern. Based on the pattern, we can deduce what type of structure is forming and adjust this pathway as needed. So, so that's kind of the first demonstration that in principle, one can build three-dimensional superlattices. This is a very simple structure, body center cubic, which we show by the time. And again, I'm not going to discuss more. There is a lot of science goes into how to analyze pattern and so on, but you can just believe me that this is something which is essentially allows us from these measurements to obtain information about the structure. So this type of idea can be applied not only to gold particles, as I showed on the previous slide, but this idea can be applied to particles of different kinds, for example, to catalytic in form of palladium, magnetic iron oxide, fluorescence quantum dots of different kinds, cadmium selenide, cadmium telluride, and plasmonic particles like gold and silver. So what is required is essentially to have a different chemistry of attachment to DNA, and this by itself will land in a different grafting density of DNA particle surfaces. So we did detailed studies how one can tune those parameters, essentially, for example, relative size of the particles, grafting density, and so on, and how it's going to lead to a different type of structural organization from crystals to weakly ordered into clusters. And this is main parameters for particles, for example, of the same size shown here. It's a number of grafted DNA in one particle and a number of another. From material point of view, this approach is essentially gives you ability to have binary mixtures in different types of organization where you can pick up functional elements, for example, catalytic particle and, for example, quantum dot. And so one can kind of mix and match those components to build superlattices of two different, functionally different components. And through the design of DNA shell, we can tune distances between the particles in the regime from 10 to sub 100 nanometers. So that's kind of allows to diversify also the structures by changing, for example, their relative size of particles. Again, I'm going to go into all the details, but we have here, I'm showing just all these studies where we have done kind of to demonstrate those details. What I wanted to stress, however, despite all this ability, we still will be quite limited in a way what type of superlattices which we can form, because in the core, it's a kind of a contact interaction, so which is provided by DNA encoding, and so we can get to different type of structural organization, for example, changing particle size. But in reality, if you have some particular types of particle, would you like to change particle size if you want to maintain their functional performance? Probably not. And the underlying problem is with isotropic potential, it's difficult to obtain complexity in structural organization beyond certain point, because at the end of the day, it's difficult to control who will be your neighboring fourths or fifths and so on coordination sphere. And so at some point we saw that it makes sense to consider a kind of a different type of approach. And when we're using not only kind of isotropic interactions, but we're using anisotropic interactions between the particles. So this idea kind of shown schematically here. And so what I want to kind of to stress, it's unlike asking only questions, what will be particular structure which will form if I do such and such, which I would call a direct problem in self-assembly. We want to develop an approach which essentially allows us to solve kind of inverse problem. An inverse problem is, this is the structure I want to get for a variety of considerations, because maybe this particle, this organization has certain kind of optical behavior of material, or for whatever reason, right? And ask question, what I need to do in order to form this type of structure in this particular approach. So the approach works in the following way. We're not touching particle because, as I stress, the kind of functional elements which we don't want to alter, but we can introduce anisotropic building blocks, which essentially connector between the particles. And so in normal circumstances, due to the entropy, it will be phase separation between one type of block and another. However, this block is encoded with DNA, is shown kind of with the gray strands, which complementary to the yellow strands in this particle, which kind of are shown here. So in some sense, you can envision this gray block will, through its kind of geometry of connectivity, will impose certain local organization between the particles. And so now one can say potentially this local organization can lead to the formation of superlattices which has similar kind of motif to the local organization. In principle, it can also lead to the disorder. You know, there are all possibilities, but if they're going to go to crystalline, it will be certainly the connection between the type of the lattice which will be formed and the geometry of this element. And so if, you know, we're smart enough, we can essentially program what type of organization will form here by adding only this block and not necessarily changing anything on a particle. So we explore this idea in a different scenarios, and I'm not going to tell you different things, but many other things, but I'll just give you a couple of examples. But you know, one of them I wanted to stress, which I'm not going to show, that we can use even other type of inorganic particles which has shape, for example, cubic particles or octahedral particle as this type of building block. But it will be still limited because you need to get some particular type of particle shape, which is kind of now possible to get different particle shape, but still not completely designable. So from this point of view, we started to, and we demonstrated it's possible, but as a greater and kind of more enabling from engineering different type of structures, we started to utilize a different type of approach, which I'm showing here. So this approach is based on DNA origami. In DNA origami, it's a kind of object one can fold and create essentially out of single-stranded DNA, which it can be folded in a particular type of organization. So you can envision that DNA is a heteropolymer, which has different motifs associated kind of with sequences. And so the motifs which shown, you know, for example, here with the same color essentially represents this kind of encoding, and we can bring those motifs next to each other by providing kind of staples. And so depending on what type of staples we connect, we essentially bringing those which are elements which that are far on a linear dimension next to each other, and that's essentially promote the folding. So by to make it simpler, by adding particular set of staples to the same DNA scaffold, we can essentially induce formation of the different types of three-dimensional, two-dimensional organizations of DNA in shape structure. And this is example, you know, I will be specifically talking about octahedra quite a lot, and specific examples this can be done. And so the great capability which is offered by DNA origami, for example, allows us to form octahedra, as you can see here. This is a cryo-TM reconstruction, the site of this octahedra cage, approximately 30 nanometers. So now the idea is essentially using DNA origami as kind of underlying design scaffold. You can use it to position particles, for example, to see how we can control local organization of nanoparticles. So here is the point that the attachment of particles to the DNA origami we can control through the DNA which placed at the vertices, and they complementary to the DNA on the particle surfaces. And this is, again, this is reconstruction from cryo-TM shown here. If all particles will have the same, and basically the same size and the same recognition, and all vertices are actually also kind of has the same type of complementary DNA, that will lead to this type of cluster. If, however, we deliberately design different type of strands to be different vertices, and we use three particles, 10, 15, and 20 nanometers gold, we can form, for example, up to us, depending how we encode those vertices, non-chiral or chiral organization of particles, because particles are plasmonic. We can verify, I mean, we can explore that they indeed show, for example, chiral optical activity for the chiral structure. So recently we kind of more generalized this type of approach that in principle you can create a variety of complex kind of clusters, and this is shown where we have a DNA mesh ball which contain 42 vertices, and you can pick and choose any of those and encode essentially different positions of the particles. In different particles can be in different places. Again, I'm not going to show all the details of this, but I just wanted to stress that it gives really ability to prescribe where particles will be positioned on this type of mesh ball. It's approximately 55 nanometer in diameter. And I'm just showing here one of the examples where we form helical structure. This is our design, and this is essentially what is reconstructed from cryo-TEM.
5 minutes.
So,
5 minutes.
Okay, and this is essentially gives us a possibility to build this type of cluster and verify. So I'm kind of half through my talk, so somehow I'm seems to be slow in the morning, at least in the morning on West Coast. So I will probably skip a couple of things just to show you something which is quite important for creating materials. And so let me just focus on this point. So one of the challenges in self-assembly, how you decouple self-assembled process from material property. And so here, the example of, we kind of generalized the project we have been developing and using frames to building different types of structural organization, where essentially you can say my lattice will be fully determined by the balance of the vertices, which is determined by itself by the underlying geometry of the object, and that allows us to build variety of different type of lattices simply by coming from different types of building blocks, and they connected to each other through vertices. And by placing different types of nanoparticles inside, we essentially create different types of materials. And so I will show you just that in reality that it does work. We can verify formation of a different type of lattices using, for example, small angle scattering and this is kind of structural organization which I'm showing here. It's a result of this. It can be while I don't have time to discuss it, we can place plasmonic particles, quantum dots. We can explore optical property of these materials. And what is important also is that while DNA is a relatively gentle material, and so you wouldn't be able to make material for broad application using this, we develop approaches how this can be converted into a fully silicated structure with incorporated nanoparticles as it's kind of given by those type of approaches which I discussed. And so in this case, it becomes three-dimensional replica, which has to be the way how we form it using DNA. And so this type of structure now can withstand above 1,000 degrees Celsius, high pressure, high radiation, and so on. And I wanted to stress it does preserve all this kind of porosity and allows us to preserve all architecture control, which given kind of by DNA. So we show that this type of approach, for example, can be used to create three-dimensional superconducting structures where we can code in collaboration with Professor Sharun from Bar-Ilan, niobium on the top of this, and essentially to form kind of three-dimensional superconductor nanostructured materials. We also demonstrated that this type of approach can be used for organizing proteins, where I'm showing here the example of streptavidin, which has been kind of placed specifically in this case, and this building block now have been assembled in three-dimensional structure. We also recently showed that this type of approach can lead to not only creating protein lattices, but also those proteins can be fully functional within those lattices. Example, using ferritin carry iron, and so this is don't have time to go into details, but we can build different types of lattices depending how we encode the connectivity between octahedra, and we can release iron while it's in superlattices, ferritin in superlattices, and monitor and characterize this process. So the last point, which is where I'm gonna finish, is asking this question that what I showed you, that it's a kind of like a complicated object which has kind of valence type of interactions, and now we also can utilize all these bonds to build kind of deliberately more complex structure. I'm finishing already. I'm just gonna, for example, to show that this type of approach where we have particles with different valences and different kind of color interaction lead to formation of arbitrary complex objects, as shown here, low resolution version of Leonardo da Vinci Vitruvian Man, as well as we can build kind of more higher complexities through the more complex encoding, and it's kind of what we're developing now, how to those approaches can be utilized for building three-dimensional structures where each object can have a high degree of encoding. So within this, I would like to finish and basically summarize what I showed. It's a pathway that we can prescribe going from, DNA object in a very well-defined frame, which can integrate nanoparticles or proteins in something which we call material voxels. Those material voxels becomes a building blocks for creating larger scale organizations through encoding how those building blocks recognize each other and forming materials. And these materials now can be transferred fully into its inorganic shape. And if you would like, this is our recent review where we have been discussing kind of designer materials using self-assembly approaches. And so that's pretty much what I want to stop here. Just want to acknowledge people in my group without whom it would be impossible to do, and many collaborators over the years, and support from different fundings. Thank you.
Thank you very much, Oleg. Questions? Oleg, maybe this also takes us to the next part. How do you see this field evolving in the next 20 years? So where do you go next?
I can see how it's evolving in the next 5 and 10 years. In 20 years, maybe we'll solve all problems and we can switch and do something other. But realistically, or maybe optimistically, I would say that I can see, first of all, I'm looking from the point of view that there's definitely a need to go to the beyond what three-dimensional printing can give you today. And so what I see, it's establishing platform for a digitizable self-assembly process, or where we can essentially create complex type of organizations, again, not only using nanoparticles, but down the road also using proteins. And I showed some of those examples, and we have full ability to prescribe the formation of the structure. So it's kind of one aspect. The second aspect, which I did not have time to discuss, is go even beyond what is possible to dream today, creating materials which we can control the dynamic behavior of this material. We can not only form some particular architecture, but also have ability to transform from one architecture to another, and so on. And so that's probably have a 20-year horizon more. So that's kind of, those are probably 2 main directions that I can pinpoint.
Great, thank you. So if there are no more questions, let's thank Oleg and thank all the speakers of the session. Toda rabba, thank you very much. And we're going to carry on to the roundtable discussion. This is going to be moderated by Reshev Teda, and we thank him again for all his contributions. Reshev was actually an IVS president, right? When was that?
Oh, a long time ago, 20 years ago, 25 years ago.
Okay.
25 years ago.
Thank you again for all your contribution over all the years. I'm sending the questions again in the chat. So we've asked the panelists. Panelists are the invited and keynote speakers to say their opinion. And I'll leave the floor to Reshev. Reshev, bavakasha.
Thank you very much, Chagai. It's my pleasure to host this discussion. And the first question, of course, that Chagai has asked is, where do we go with nanotechnology and nanomaterials in the next 20 years? It's hard to predict and say things, but I think that for next 5-10 years, one can have certain ideas and certain directions that one can conclude. And let me start by saying, first of all, in my view, one of the key issues is to improve our ability, and not only to synthesize materials, but also to measure materials, but nanomaterials, one particle at a time. And measure is very global. 15 years ago, I was able to start with Yifat Kaplan-Ashiri to measure the first mechanical properties. Actually, 20 years ago, Daniel Wagner from my department was able to measure the properties of individual carbon nanotubes, some of their properties, but of course there are much more than that. And the question is, what do we see as a future tools, and how shall they will evolve in the next 20 years or 10 years from now? And I can give you an example. For example, if you look on quantum dots or nanoparticles, you can measure easily a materials, a single nanoparticle, if you have photoluminescence, because photoluminescence is very easy to detect from a single nanoparticle. You collect it with a large numerical aperture objective, and you get the signal. But if you think of, for example, if you want to measure the absorption coefficient of one single nanoparticles, it's quasi impossible. It's almost impossible. It's very, very difficult. And there are many, many examples to this kind of difficulties that we face basically daily. So I think people should talk and discuss it, and I'm ready to listen. And maybe before that, I'll give another example, just to give you a hint of the difficulties that we and others face. So if you take molybdenum disulfide or graphite or boronitride, 2D materials, one layer at a time, you look in the electron microscope, you can view them very nicely. You can analyze them even atom by atom, maybe. Nowadays with EELS or with EDS, the performance has been improving so much. But let's say you start to curve these structures. And I'm not speaking about my own experience of multi-wall nanotubes, even single wall nanoparticle. You want to curve it, or even to take it a 3D structure of a quantum dots. It's not trivial by electron microscopy, because there are always interference from the neighboring atoms. And this is another example of how difficult is it to characterize materials on a nanoscale. And this is two examples for difficulties, but there are many more cases. And perhaps if you want to measure, for example, the mechanical properties of a single nanotube or a single nanoparticle in a polymer, it's not trivial. By all means, it's difficult. You use Raman, but this is an indirect hint. You want to measure the mechanical properties of a single nanoparticle in a polymer. It's not trivial. To measure the surface energy, to measure how much strain goes from the polymer to the nanoparticle, it's not easy at all. So I think this is one of the things that we need to improve better and to improve on ourselves in order to be able to answer questions more precisely and get different ideas on their applications. I'm ready to listen to other people in the audience.
Yeah, I can talk a little bit about my experience with watching in the past, actually, situations where it seemed that, let's say, the bottom-up approach, right? We are comparing usually, right, the bottom-up with the top-down. So far, right, in microelectronics, the top-down has been doing fantastically well. So one interesting example was hard disks. At around about I think the turn of the millennium, something like that. So people started making magnetic nanoparticles, borrowing the methods they brought from quantum dots, for example, to make them more uniform and more variety of materials. And there was a point where and of course, I mean, the perfect magnetic storage medium has been always single domain magnetic particles. So already right from the magnetic tapes, I mean, from the 60s and 70s, people were using actually colloidal nanotechnology for magnetic storage. So So at around 2000, came the opportunity to make hard disks even better by using colloidal magnetic nanoparticles compared to the standard thin films that they use to evaporate, for example, I don't know, combinations of cobalt and chromium to make islands of cobalt. And unfortunately, this didn't work good enough. And I think there are no products. This was an interesting example, but it didn't really work. This could really make a very strong impact on magnetic storage if it would work. So there were many technical problems with that.
Can you tell us some of these problems?
Yes. So the ideal magnetic storage material is iron platinum 1-to-1, right? But so if you get the right phase of the 1-to-1 iron platinum, you can get even a particle that is as small as 4 or 5 nanometers to maintain the magnetization at room temperature for a long time. So this is much, much better than iron or cobalt, of course, compared to iron oxide. But to get this phase, you need to, the colloidal synthesis gives you a random distribution of the iron and platinum. So you need to anneal it at high temperature. So this made things more complicated, together with the fact that you need to get very uniform films, which also was a little bit difficult in spite of all kinds of techniques like, Langmuir blowjet and many other ways, spin coating and whatever. And people also looked at Glucopolymers as a way to pattern, So there was a huge effort. Seagate at that time and IBM invested a lot of efforts to try to make it work, but this particular example didn't work. And then later on, actually, recently, I was working myself also on this problem of the ITO replacement by metal nanowires, which was another way of colloidal method to try to replace very traditional thin films, which also, eventually didn't work. There were several companies formed and some products were there, but nothing, some demonstrations. But eventually, ITO still is the king. But still, it doesn't say that in the future, the quantum dot example for the TVs, screens, and so on, this worked. But I would say that it's relatively a niche application, but still there are some particular examples that probably could work better.
Joseph, can I say something about the characterization? I think you were very right about the challenge there. And what I wanted to maybe emphasize here is that there are always trade-offs. If you want to do spectromicroscopy or microspectroscopy, so combine the images or the ability to measure both the structure and the properties in a way, which is, I think, the direction everybody tries to go now. And take, for example, the electron spectroscopy, you can get amazing results, amazing, but the huge or the resolution is lateral. So actually, in depth, you collect over large distances.
Yeah. And
on the other hand, if you look at the technique like what I do, XPS, the surface it's a huge sensitivity to the surface with very poor lateral imaging, and so it goes also with the properties that you can get in each technique. So one direction which looks the way to go is combine a number of techniques, because the trade-off will be always there. So the question is whether you can put in one position the sample and then have a lot of ways of detections and in excitation also to get many types of information all together. And in contrast to measurements that you do in different chambers, different techniques, and then so many parameters are changed, and it's so difficult to actually get to the conclusions. I think that this is the challenge, to make a machine that is multi-function. Right?
Guy, let me comment on that. In the past, I recall there were machines that were able to measure Auger and XPS together on the same machine to get just to answer the same question in a way, namely to be able to probe single nanoparticle or single domain and measure it with Auger and then go with XPS and try to characterize it more and lateral with loss of a lateral resolution. I think that nowadays people are decided to separate the two machines and to make them, to try to improve each one of these techniques by itself, rather than trying to combine them together and make them a unified characterization tool. Isn't it?
It's correct. I think that part of the loss of interest in Auger is not justified, but there are technical issues of how to actually optimize the Auger capabilities and the XPS capabilities. It's not easy to do that.
Mm-hmm.
So, but this is just an example, okay? But in the electron microscope, people do today the cathodoluminescence and the few different detectors. So there are attempts in this direction.
Yes.
But I think it should go much, much further.
Mm-hmm.
And I believe that as things are becoming more automated and machine learning is coming into it,
yes,
things will become easier and easier. So probably, I think that this is anyway the promise.
One way to improve on our capabilities is, of course, to make a laptop synchrotrons. If we could make a laptop synchrotrons, we could think of bringing, let's say, a synchrotron to every research lab, rather than running to big centers and being able, because if we could have the X-ray a few orders of magnitude stronger in our X-ray diffraction, we could go to much smaller areas. And I guess the same for XPS. If you could make a synchrotron radiation, a kind of synchrotron radiation on laptop synchrotron radiation, and you could put it in a room and do XPS with a few orders of magnitude stronger irradiation, you could skim the beam and get to a single nanoparticle. This is one of the goals, I think, of the future development in instrumentation.
Reshef, I think Tamar wants to say something.
Yeah, I wanted to add to the discussion that you and Hagai mentioned, and I agree with you. And to add an additional challenge to that, with the huge development of three-dimensional microscopy, Oleg has also shown us a beautiful result from his work. It would be great to be able to characterize specific properties within the three-dimensional structure. So that's one comment I had. I would like to add additional challenge more on the application side. And I think that sustainability, combining that with nanotechnology, nanomaterial is one of the major challenges we should think of, both how to use our development of nanomaterials for better processes, and also when we in the industry are working on development of semiconductor manufacturing, magnetic media, everything that was mentioned here, how can that be done in a more sustainable way? So I think that needs to be more in our focus.
Just a comment, Tamar, you actually hit my second part of the discussion, and that will be the next 10 minutes. But okay, Hagai, please comment first.
Yeah, actually, I was going to the next part because we have 4 minutes until we have the next plenary session, right? So we have 4 minutes. So if you want to introduce the second part, and we'll...
Okay, so I will state it the following. As Tamar noted, I think it's the future of materials will be tightly bound to our ability to control the environment and sustainability. And one example, which also relates to my previous discussion, or the previous part of the discussion, is catalyzed. You have a big nanoparticle which is probably made of hundreds, if not thousands of atoms, but not all of them are active. Only few of them are active, or few sites are active. And you want to understand this site, and you want to optimize it so that the nanoparticles will be more active. And so this actually requires, on the one hand, more advanced characterization tools, and at the same time, improve our ability to crack split water to hydrogen and oxygen, or carry out any other or Fischer-Tropsch reaction, ammonia production with catalytic nanoparticles, which will be more active and more selective. So I think this is a very, very important issue.
Reshef, I'm sorry to be the very bad guy here, but we have the plenary talks about to start in 3 minutes here. We're inside the break, so I would suggest, if you all can, to sorry, Mechila, Reshef, for that. I assume it was a very interesting and live discussion, but we have to move on with the 2 plenary talks. So if you can reconnect to the plenary, I would appreciate. Thank you.
Okay.
Thank you.
So I want to conclude this session by saying that we surface several issues in the next 10 or 20 years of research and development for nanomaterials and nanotechnology in general. There are many, many more questions. And if there are no more further questions and comments, let's go on to the plenary.
I have one.
Please, go ahead. Micha.
The theoretical part of nanoscience or nanotechnology you didn't mention.
All right.
There have been a lot of developments in computer simulation, DFT, etc., to, for example, predict the compositional structure or chemical order in nano or alloy nanoparticles. And we are able now to predict nano phase diagrams of nanoalloys, certain nanoalloys as function of the size. And such information about the thermal stability of nanoparticles is very difficult to obtain experimentally, while theoretically the situation is quite in progress and maybe superior to the experimental ability. So that's all. I mean, theoretical modeling and simulations are quite in progress in this field and are very promising. Thank you.
Micha, I fully agree with you, and I can only thank you for your comment. I think it's a crucially important in the development evolution of nanotechnology, and I thank you very much for this concluding comment. Thank you very much, Micha.
Thank you.
So thank you all. I've posted open positions. Igor Rachinov suggested that we post this if anybody's looking for positions at Princeton University to go there as a postdoc or something like that. You can look up the link. So thank you all again. Thank you, Reshef. Thank all the speakers. Toda Rabba, and let's move on to the plenary session just on time, 3:30.
Bye.
Thank you very much.
Thanks, Chagai.
Thank you very much.